All right, I'm going to go ahead and start. Just a reminder, next Tuesday we have a fun, exciting event that's going to be taking place. Yay! Test. Boo. Yeah. Yes, it is actually on, so I am recording this now. So remember, if you haven't signed up for a test by this point, you're really going to be stuck with like the worst times, like the 8 o'clock in the morning, have to fight traffic into the medical center times. So go and make sure you signed up for the exam. I've actually seen where students thought they actually signed up, but they forgot to accept or click or whatever. So just go back and double check to make sure that the time that you thought you signed up for is the actual time that you signed up for. Secondly, Mu Delta is having their first general meeting tomorrow. So that will be in the Honors College Room 2112, and then that time is 7 p.m. to whenever. So that's Mu Delta's announcement. If you didn't go to the Taco Cabana meet and greet, well, they won't know who you are, but that's okay. They're still waiting to see who you are. SASE, Society for Asian Scientists, or of Asian Scientists and Engineers, is having their first general meeting next Monday, the day before the exam, because I know you're going to attend that one. Right? They might have all the answers to the test. Anyway, so again, 7 to 9 in D3W205. I have no idea what building that is. So that's the engineering building. See, that's why I don't know. Yeah. But notice the key thing here. So if you need a break from studying, what do they have? They have free food. So they'll be happy to fuel your needs as you're preparing. All right. So just remember, next Tuesday is your exams. My tests are not that hard. Quit letting people scare you. Just telling you. What we're going to talk about today is we're going to talk about hormones. We're going to talk about the endocrine system in a general sense, and also we're going to talk about some specific hormones in a specific sense, just to see how they work. All right? And then as we go through the year, we're actually going to come back to a lot of these hormones and talk about them in terms of real action. So the depth to which we're going to go is that's the depth at which I'm going to test you. All right? Just had a person come up with a question, say, I saw a question on the practice, on the Mastering A&P, and asked me all sorts of stuff about pepsin. Do I need to know about pepsin? I said, no. You don't need to know about hormones we don't talk about. So just keep it simple. Now, as we were talking about on Thursday, we said that hormones can be a little bit complicated because there's all sorts of rules that basically break all the rules. Right? They have all sorts of features, like if you have an endocrine organ, it doesn't just secrete a single hormone, it can secrete multiple hormones. And it just gets kind of confusing. But what we're going to try to do is we're going to try to break away all this confusing stuff and keep it really, really simple so that you can look at this stuff and say, oh, this isn't as hard as people make it out to be, even though there is a full course about endocrinology. All right. Anyway, the way hormones are classified is they can be classified either by their source of secretion, their mechanism of action, and this is the type of receptor used, so we've kind of already learned this. Its solubility in water. Have we learned that already? Yeah. Or by their chemical classes. And this is really what we're going to do, is we're going to focus here on the chemical classes, and then we're going to come back and we're going to see how they apply to these two things. All right? So what we have peptide hormones, or what we refer to as protein hormones. We have steroid hormones. Everyone's heard about steroids. And then we have the amine hormones, which confuse everybody because they don't fall into either of these two other classes. So let's just kind of walk through first the peptides, and then we're going to go through the steroids, and then we'll deal with the amines, which, again, no one likes. So we're going to look at first synthesis. We're going to look at how they're stored and how they're released, how they move through the body, and then ultimately their mechanism of action. And while these slides look very, very busy in terms of words, they're not hard. First off, proteins. It doesn't matter if they're a hormone, if they're an enzyme or anything. Proteins are all made this exact same way. Basically, you're going to start off with a large molecule. This large molecule is called a pre-prohormone. This is going to be done in the endoplasmic reticulum. All right? And so you're going to take this large pre-prohormone and you're going to start processing it, and you process it in the Golgi apparatus. Aren't you glad you took Biology 101? Yeah. All right. And then from there, now you have what we refer to as an inactive prohormone. All right. And here, this is where you're going to be sorted. Remember, the Golgi apparatus is like the mailroom of the cell, and so you're sorting molecules all over the place. These prohormones are going to be sorted and tagged and placed into vesicles. And here, once they're put in the vesicles, they're going to be modified one more time. All right, and here we're putting them into their active form. And this is where we actually refer to them as a hormone. And we're going to keep them in these vesicles until we get a signal. The signal being, cell, release your hormones. And at that point, you get endocytosis. All right, so we're going to release both the hormones and the peptide fragments that are left over from that processing, and that's going to be released out into the bloodstream. Now, because they're proteins, or peptides in these cases, because we processed them down from this large molecule, this large protein, is peptides, by definition, are water soluble. So when they go into the bloodstream, they just happily float along like every other plasma protein that happens to be around. They just move and float around. And so they're very, very happy. Now, these peptide hormones do have short half-lives. This is going to be true for a lot of hormones because you don't want the signal to be just circulating around. Right? If you have a signal circulating around, that means that signal can be amplified when it binds to a receptor at the worst possible moment. Right? All right, let me put it into another way to make this make more sense. You want to regulate very carefully what signals are available to signal to the cells and telling them what to do. If you don't regulate that in a fashion, you can have very bad things happen. I'm going to use an example that does not have to do with this type, but it's one that makes the point. Have you guys ever heard of something called breast cancer? You have, okay. What is one of the forms of breast cancer that we all have heard about? Estrogen responsive, right? In other words, what happens is that the overexpression of estrogen receptors basically allows certain tissues to misbehave. So just the presence of the estrogen allows for this tumor to grow. All right. Now, I'm not trying to scare you ladies. Don't freak out about breast cancer. That's not the goal here. But the idea here is if we can regulate and control the signals, then inappropriate things won't happen. And so by having very short half-lives of these molecules, they can be destroyed very quickly. And then if you need the molecule again, what do you have? You have a signal that says go and release the molecule. Now, typically, these molecules, because they're hydrophilic, they love water, they're going to bind to cell surface receptors. Right? Because they can't penetrate through the plasma membrane, the only place that they can actually bind is going to be at the cell surface. And so, generally speaking, they're going to be acting through second messenger systems, which is why we learned about them. Yes? Sure, that works. Except what if you have a tissue that's all of a sudden actually expressing receptors shouldn't? I mean, so when you're starting to produce a whole bunch of molecule, right, you can actually downregulate in response to that. But if you have a tissue that is behaving inappropriately, in other words, it's not being regulated in the proper way, and it just starts expressing a receptor by virtue of it misbehaving, then that's where you're going to see stuff like that. Right. Right. So really when you think of tumors and you think of cancer, side issue, don't write this down. When you think of cancers and you think of tumors, think of cells not behaving the way they should. And it can be any multitude of ways, whether it be the misexpression of receptors or inappropriate responsiveness to a signal, even if there is a downregulation, or it can be an inappropriate second messenger system or any sort of signaling cascade, or just metabolic activity inside the cell. So all sorts of fun, nasty, horrible ways to die. Oh, I'm sorry. To do that, yeah. Absolutely. So, and in fact, some of these can be used as a way to measure the degree of hormone that's available in the system. Yeah. Well, I mean, so they'll be destroyed more or less the same rate, but what you can do is you can actually, take a blood sample and look, for example, like with insulin, you can look at C-peptide, which we'll see in just a second as an example, not something to memorize, please. All right. Now, because you're using second messenger systems, what you're going to see is that there is a very short period between the moment that the actual hormone binds to the receptor and the response in the cell. And remember why that occurs, because we already have a pathway in place, and so all you got to do is just start the first molecule in that pathway and you get that cascade. So the response is just going to occur very quickly because we already have things in place. Now, For those of you who need to have pictures, this is what the pictures show you. And so you can see here we're making our pre-pro hormone. It gets modified, gets sorted, sent in the Golgi apparatus, sorted up in the Golgi, and then modified again. And what you're left with is your active hormone plus peptide fragments, and then that stuff gets released out into the bloodstream and goes on to your target tissue. Pretty straightforward stuff. These are some examples. Do not memorize these. I'm not going to ask you how do you make TRH, for example. But it shows you, here's the pre-pro hormone. And when you cleave that sucker, you get 6 TRH molecules, thyroid-releasing hormone. You get some other peptides, and then you get the signal sequence that gets thrown away. Here's a fun one, pro-opiomelanocortin. What you get is ACTH, lipotropin, and beta-endorphin, all from one molecule. So it's a 3-for-1 deal. All right? There's insulin, actually proinsulin, and then it gets cleaved, and that's what the insulin molecule actually looks like in terms of a peptide fragment. And so you can see it's a peptide, and there's your C-peptide over there. That's one of the things that you can measure, see how much insulin you're actually producing. So you can see that there's a processing process, or that sounds stupid, there's a processing mechanism in place to create these peptides from much larger particles. And again, these are going to be water-soluble. And because they're water-soluble, but they bind to an assortment of receptors, and I'm trying to make sure that you're not just thinking, oh, it's always going to be a G-protein coupled receptor. It can act through tyrosine kinases. It can act through a G-protein. It can even act through a ligand and a ligand-gated or chemically gated ion channel. So any of the mechanisms that we described, these are all fair game. All right? But you understand, if there's a receptor on the surface that binds that particular molecule, then I can activate all sorts of different types of events. Steroid hormones, on the other hand, are going to be synthesized in the smooth endoplasmic reticulum. And it's done so through cholesterol that has been stored inside the cell. All right? And so there's a series of enzymatic reactions that I think I've actually not put the different pathways in because people try to memorize them. And that's something you get to do in biochemistry. You don't have to do it for this class. And what we have are very specific enzymes that are responsible for producing very specific hormones. All right? And so each hormone is going to be produced by a specific cell type in the body. All right? So our organs are specialized to produce specific steroids. Now, steroids are lipids because they come from cholesterol. If they're lipids, that means they are lipid soluble. What that means is that they don't bind to cell surface receptors. They bind to nuclear receptors. They penetrate through the plasma membrane. And again, we're ignoring the exception to the rule. All right? Now, because they are lipophilic, they do not like water. That means when they are released from the cells, and that means when they're released, the moment that they're made, they're released. All right? Because you cannot hold on to a lipophilic molecule. You can't put it in a vesicle for storage. Right? Plasma membrane, is that going to contain a lipophilic molecule? No. So the moment you make the steroid is the moment that it gets released. And it goes into the bloodstream, and the first thing it does is it panics because everything around it is water. All right? And so what you have to have is you have to have transport molecules, binding proteins, that actually can pick these things up, protect them from the water, and then allow them to travel around through the body. All right? Now, you have very specific carriers that do this. This example I threw up here is corticosteroid binding protein, but you have them for all the different types of steroids. There's an androgen binding protein. There's an estrogen binding protein, so on and so forth. All right? But you can also have non-specific carriers like albumin. If you don't know what albumin is, go home, take an egg, crack it open, and poke a little bit at the white stuff, not the yolk, the other stuff. That's albumin. All right? Notice when you touch it, what does it feel like? Sticky. All right? I mean, go play with it for a little bit. You know? It's sticky. All right? It's capable of binding a whole bunch of different things. All right? Now, the benefit of these carrier molecules is that because they protect the steroids, what they do is they actually extend the half-life of the molecules. In fact, they protect them so well that the molecules basically can stay in circulation for quite a while. But because they're also bound, they're typically, we refer to them as being biologically inactive until they are released from the carriers. And so the way that we can actually look at the active molecule is looking at the ratio of bound molecule to unbound molecule. And how is this all regulated? Through the law of mass action. Right? Now, here's the thing. Law of mass action can become very, very confusing. And really all it is basically saying there's a perfect ratio between molecules. All right? If you look at an equation, for example, a chemical equation, you know that there's a nice balanced equation. If you get this side versus this side, it should be balanced. Right? So if I take something from this side, it's going to shift the equation in this direction. Does that make sense? Right? And if I take something from this side, it's going to shift the reaction in that direction until we reach balance again. That's the law of mass balance. All right? And so you can think of it in terms of bound molecules to unbound molecules. If I have a perfect ratio, and the ratio doesn't have to be 1 to 1. It can be 100 to 1. But that's the perfect ratio, whatever it is. And if I remove something from this side, that's going to create an imbalance that's going to cause molecules to move in this direction. All right? How do we... And this bound to unbound is actually something that's going to be maintained within the bloodstream, even though that makes the unbound molecules very unhappy because they're in the water. So they're going to spend some time trying to find their receptors, trying to find those cells. But imagine, for a moment, a steroid molecule disappearing from the blood. What's that going to do? It's going to shift the reaction to cause another molecule to become unbound, and now you have a molecule that's available for interaction one more time. All right? We're going to come back to the laws of mass balance over and over again. I've got some great examples. We're going to talk about M&Ms and Oreos. And I promise you, you'll understand this principle if we don't by the end. Yeah.
You said is there a difference between the law of mass action and the law of mass balance?
No, it depends on who's writing. I swear to you, it really is. Yeah, it's basically, one is actually a principle in terms of how, things relate in a biological sense. Other is really kind of a chemical reaction type thing. But it's the same principle. All right. Now, the way that steroid molecules actually interact is they have to bind to our intracellular receptors. Now, again, once they get inside the cell, you're going to come into contact with a watery environment. And so again, the idea is that these molecules are very unhappy. So they're either going to bind to carriers inside the cell, or they're going to quickly find their receptors and bind to it. Now, the receptors, these intercellular receptors, sometimes go by the name of nuclear receptors, or typically intracellular receptors. But back in the day, it was called nuclear receptors because that's where all their action is. But a nuclear receptor spends as much time in the cytosol as it does in the nucleus. It's just when it gets bound up, that's when it moves to the nucleus. And when it goes into the nucleus, this complex of ligand and receptor is now a transcription factor. And as a transcription factor, it can turn on or turn off genes. And when it turns on genes, we're making new proteins. And when we make new proteins, we're changing the functions of cells. If we turn off genes, are we changing the function of cells? Yeah. But we always like to think in terms of turning things on. But you can turn things on and turn things off at the same time and still get a new effect. All right? And so this turning on and off of genes resulting in new proteins results in in a relative sense, takes much, much longer than turning things on when you already have all the proteins in place. Does that make sense? Right? So what we have to do in order to change the function is we have to make our proteins. If this isn't making sense to you, which is it easier to do? To go and take a piece of cake off a plate, or to go and bake a cake so that you can take a piece off the cake? That's right. It's easier to go eat that cake. Forget the baking part. And this is what Hostess figured out. Anyway, all right. Now, as I said, and this is a parenthetical, I will not test you on this, but you're going to take other classes where you're going to learn that there are membrane-bound receptors that can be bound by these types of molecules, these steroid molecules. That's okay. I'm not going to test you on that. I just wanted to throw that up there. So that is a parenthetical for this class. What I wanted to show you here is there's cholesterol. All right? And what happens is all we got to do is make some minor modifications to this cholesterol molecule, and these little minor modifications turn into massive differences in terms of responsiveness. Let me put this in perspective for you. And I don't have it up here, but there's estrogen. I don't have testosterone up there. But just by virtue of you thinking about it, do testosterone and estrogen have different effects? I mean, here you are in puberty, and let's say you're a female and I start pumping you full of testosterone. Are you going to look like a little girl? Or are you going to look like a big girl? No, what are you going to look like? Right? And I know I'm being a little facetious here, okay? But, I mean, you understand this concept. I mean, you always think of testosterone being the male hormone. And similarly, I mean, what's the classic joke in many sitcoms and movies? I mean, what was the Adam Sandler one where he was the Final Yard, I guess, where they remade the Burt Reynolds movie? Longest Yard, thank you. And remember, what did they do? They snuck in and they replaced one of the guards' vitamins with estrogen pills. And so, it's a classic joke. He starts, behaving very femininely, and it's like, that's not what happens. But again, you can think of it in terms of, all it takes is just a couple of changes. I mean, look at these. I mean, there's aldosterone. Aldosterone and cortisol, very different molecules. This one regulates water retention. This one regulates stress responses. And all it takes is a couple of side chain changes. So minor changes, minor differences result in major biological changes in response to or with regard to the steroids. Now again, just showing you the mechanism of steroid action. Remember what we're doing here. We are bound to our carrier. We get released because there's a perfect ratio between those two states. The one that's released can actually enter into a cell. If there's a receptor for that cell, it will bind to it, translocate to the nucleus, cause us to make mRNA or not make RNA. And ultimately what we're going to do is make new protein. And then this is the other new way that we're just going to ignore for right now. Ignore for now. Okay? So nothing new on that. The third type are the amine hormones, and these are the ones that just screw everything up. All right? Because the amine hormones are simply put, are taking amino acids and modifying them, and then they behave as hormones or signaling molecules. And there's nothing wrong with that except that one behaves like a peptide, the other one behaves like a steroid. So it's just like, you've got to actually memorize which one's which. All right? So the two amino acids that we use are tryptophan and tyrosine. Tryptophan results in the formation of a molecule called melatonin. All right? The tyrosines form the catecholamines and thyroid hormone. All right? Now, the catecholamines, they act like a peptide. They bind the cell surface receptor. What are the catecholamines? Adrenaline, noradrenaline. All right? And so you can think of, all right, if I'm a peptide, I behave through a second messenger system. Okay? And then the thyroid hormone, for example, acts like a steroid hormone. So, you see, it's like the amines are the ones that screw everything up. You had this nice simple system, and now you got something that you have to memorize, something external. All right? Now, I'm probably not going to be real strict on this and say which one does which. But you might want to, just in case. I can't remember if I have questions on this or not. Yeah. Melatonin, I believe, acts as a cell surface molecule. Yeah. Mm-hmm. Now, to me, this stuff is the stuff that becomes more important, because when you deal with specific molecules and what their specific action is, we need to be putting them into context. I mean, me just coming up here and listing a whole bunch of molecules and saying this is what it does doesn't serve a really good purpose. I mean, would you agree with that? I mean, there's nothing worse than memorizing a list of things and you have no idea other than what's on the list, right? So I want to deal conceptually for a moment, all right? Because understanding how hormones behave allows us to see how these molecules work in context when we come across them again. So generally speaking, what we have is what we refer to as a simple negative feedback loop, which is responsible for regulating the amount of hormone that's being produced. So, as an example, your body is responsible for regulating the levels of calcium. And the way it does that is you have cells that are constantly sampling the blood. And when it notices that we have low calcium levels, these are parathyroid cells, what happens is these parathyroid cells are then, because of this low calcium, begin to produce a hormone, which has a real complicated name called parathyroid hormone. All right. Now, the parathyroid hormone, what it does is it travels in the blood and it goes to the bone and the kidney, and it actually increases calcium levels by basically increasing the rate at which we resorb bone. In other words, we're taking out calcium from it. It increases the rate at which we reabsorb calcium from the kidneys, so we're not just peeing it all out. And also acts on the intestines by increasing the production of calcitriol, so it's actually a multiple-step layer. Calcitriol is vitamin D, by the way. And what you're doing is you're absorbing more calcium. All right? So the consequence of all this, if I'm not peeing out calcium and I'm breaking down my bones and putting calcium in my body and I'm eating and drinking more calcium, my plasma calcium levels go up. And as my plasma calcium levels go up, that's going to be read by the parathyroid cell, which then says, you know what? I don't need to make any more parathyroid hormone. That's the negative feedback loop. All right? So what we have here is a cause and effect. The cause was calcium levels are low. The effect is produce a mechanism to stop the low levels of calcium. Once the calcium levels are no longer low, that's going to stop the effect that caused the whole pathway. So you end up going through a process of bouncing back and forth between this stage of too low and just right. So you basically kind of wobble in these two stages, or within a range. So this is kind of a simple reflex. And this is how many of the hormones in your body are actually regulated. And we're going to be looking at some very specific examples with regard to the neurohormones. Now, there are three different classes of neurohormones that you should be aware of. And if you look at this slide, there should be something up here that's terribly confusing to you. Have you noticed it yet? Yes. There's two things that have the exact same name. That wasn't a typo. That was for you to pay attention. All right? It's like, crap, there's something here that's weird. All right. The first major class of neurohormones are the catecholamines. You've heard me mention them again. This is adrenaline and noradrenaline. And they're produced by a modified neuron. We'll spend more time talking about this after the test, called the adrenal medulla. All right? And so here, adrenaline and noradrenaline are going to be released in the blood and then travel throughout your body, and they support the sympathetic nervous system in its response. If you're sitting there going, what's sympahuah? Don't worry about it. We'll get to it after the test. The other two major classes of neurohormones are both hypothalamic in nature. All right? In other words, they come from the hypothalamus. The first type is produced in the hypothalamus, but is secreted from the posterior pituitary gland. We'll talk about that in a moment. The second type of neurohormone that comes from the hypothalamus is made in the hypothalamus, if it's hypothalamic, and then it's released by the hypothalamus and acts on the the anterior pituitary gland. So notice what we got here. One is actually made in the hypothalamus and then secreted by the posterior pituitary. The other one is made in the hypothalamus, released into the bloodstream, travels to the anterior pituitary, and makes the anterior pituitary do things. So one is regulatory in the sense that it acts on another tissue. The other one is released by a tissue and has some sort of function. Now, how is it possible? Yes. Well, actually, they don't have names themselves. You just call them hypothalamic hormones. All right, because it's where it's made. Right. And that's a very good question. And in fact, when I was writing the slide up, I was like, I got to make this stand out. I got to make it kind of confusing so that you have to stop and think about it. So there's no real name. They're just hypothalamic neurohormones. That's a good point. Yeah. Yeah, well, I'm actually going to be dealing with those in just a second. Say it again. How do you distinguish one? You will see here in just a moment. Yeah, the good news is that there's not a lot of them. All right? And actually, the way that they're named actually makes it easy to learn the majority of them. The second group, the one that act on the anterior pituitary, they're easy in terms of their nomenclature. All right? But what this leads us to is first we need to understand what the anterior pituitary, what the pituitary gland is. It has another name called the hypophysis. And if you look at the the pituitary gland, it actually has two unique and separate origins. If you want to look at the picture on the next slide, there's the picture right there, as I go along. So there's two halves to the pituitary, and you can already guess which halves they are. One is the anterior, one is the posterior. One's the front, one's the back. The anterior portion is glandular tissue. It originates from epithelium. The posterior is an extension of neural tissue that comes from the hypothalamus. And so what you end up with is this structure that's terribly phallic looking, like that. And it's okay to think about it being phallic because there are reasons why we can say that. All right? But the posterior side, there's the anterior side. The posterior side is neural. This is glandular or epithelial in nature. All right? And so what we have are hormones that are going to be released from these two tissues, and they're both being regulated through the hypothalamus. All right? In regard to the anterior the anterior pituitary, here's your hypothalamus up here. It's releasing hormone into the bloodstream, which travels in the bloodstream and acts on the glandular portion. All right? So here... Well, no, so I'm just kind of... I said look at that picture so that you don't have to look at my horrible drawing. So this is the anterior right here. So what you have is you have a neural link that leads to this. I'll get to it. I told you I can't draw it. That's why I wanted you to look at the picture. All right. The posterior, on the other hand, has what we refer to as a neural link. In other words, this tissue are a series of axons that originate up here in the hypothalamus. What's an axon? It's just nervous tissue. We'll deal with it in just a moment. And that terminate down here. So let's see how this works. We always start with the posterior because it's easier and we can get it over with. All right? So what we have here is an example of one of these neurons. The neuron up there is the cell body found in the hypothalamus. It's found in two areas. I'm not going to test you on them, but just in case you see them ever again, you'll know supraoptic and the paraventricular nuclei are the two areas where these cell bodies are found within the hypothalamus. I will not test you on that anatomy. All right? But these cell bodies produce two hormones, vasopressin and oxytocin. Vasopressin has another name, antidiuretic hormone. For those of you who get confused by names, antidiuretic hormone is simply put anti-peeing hormone. Okay? Actually, this is an aside just because it's fun and I like talking about it. These two hormones are very closely related, and in some animals, they only have one, and they do the job of both of these hormones. All right, very closely related. You guys have heard of oxytocin, right? Yeah, what does oxytocin do? All right, yeah, it's contraction, but it does something else that's very important. I heard it. What was it? The cuddle hormone. That's right. It's a hormone of love. Literally. All right? That is the feeling. When you get a kiss, that feeling, that, oh, I really, really like this person, is a result of the release of oxytocin. Let me ask you this question. This is just an aside. Who do you think is more responsive to oxytocin, males or females?
Women.
Again, this is a little bit more fun. All right, we're going to put this. Who is more susceptible to? That's not the right word I want to use. Which one? Who is more prone to be in a one-night stand? A male or a female? I'll leave that answer to you all, and then you guys can interpret what that means. Huh? No, the answer really is that females are actually more susceptible to oxytocin. All right? Guys are basically love them and leave them. I mean, yes, I know there are guys who get all weepy and, sad and write poetry and stuff like that. But in the grand curve of things, the majority of males are basically love them and leave them. Women, for the most part, are not love them and leave them. I know that women try very hard to do this, but they get pretty weepy and clingy. And don't get mad at me. And it's because of the availability of these receptors. All right? Now, the reason for all that stuff, and again, this is all aside stuff, so it has to do with the ability to, create bonding pairs and the mating strategies of primates and other things. So, just...
It's not my fault.
Yeah, there you go. Yeah, if that doesn't beat you down, I don't know. You're I'm going to find you in an alleyway broken in about 6 different ways. All right. But you see what we have here is we got these two hormones that are being produced up here in the hypothalamus. These axons from these neurons travel down and they create part of the stalk of the pituitary gland. All right, and then they terminate down here, and the vesicles that have these molecules are in the posterior pituitary. And so when a signal comes along to the hypothalamus, that's going to result in a signal that causes the release of the vesicle and thus the release of the neurotransmitter into the bloodstream. All right? And then it travels on and does whatever it's supposed to do. Now, just to point this out, this cell, one cell right here, produces either oxytocin or produces vasopressin. It doesn't produce both. All right? But if you have two cells originating in the same nuclei, one can produce vasopressin, one can produce oxytocin. It's just one cell produces one hormone. Does that make sense? In other words, you can't just say, over here in the paraventricular, it only produces vasopressin. No, that's not true. They each produce both. All right? But where they terminate and how much or what they produce is only going to be one type of molecule. Does posterior pituitary make sense? Molecules are produced in the hypothalamus. They're released from the posterior pituitary in response to a signal that's going to be received up here in the hypothalamus. Yes? Yes, in this particular case. All right? So, if we're in, you can do it like, I'll try to do it like a drawing. If I can find some shots. You can think about it like this. See, got right to my perfect height. All right? So here we are. I have one nucleus coming down, terminating, and from the same location, another one. Over here is the other nuclei coming down, and I guess I'll do it like that. So we could have producing oxytocin, for example, vasopressin, and even though they're in the same area, they're producing different hormones. This one could be producing vasopressin, this one could be producing in a different location. So that was my point. They both can't produce two. They only produce one, but they can be in the same location. And then ultimately here you are down in your nuclei. So here you can actually release whatever it is. Look, it's valid. I saw that look. The anterior pituitary is where people start getting all scared and freaked out because it's like, oh my goodness, there's so many hormones, I've got to memorize 6. All right. The types of hormones that you're going to find in the anterior pituitary are called tropic hormones. Do not confuse these with tropic hormones, which is what you get when you're down on spring break. Tropic hormones, thank you for laughing. It took me a long time to think of that one. Tropic hormones, simply put, it's also, you can see the term trophic, T-H. All right, so it's tropic or trophic. Either one is correct. All right. Their job is to regulate the secretion of another specific hormone from another endocrine gland. And so the trophic hormones include growth hormone. You're going to notice how easy these are. All right, we have growth hormone, thyroid stimulating hormone, the hard one, adrenocorticotropic hormone, AC, so adrenal cortex, tropic, T, so tropic hormone. FSH and LH together are called the gonadotropins. FSH is follicle-stimulating hormone. LH is luteinizing hormone. I'm far too lazy to write these things down, so if you need to go look them up, that's fine. I don't know if they're on another slide. All right. The only hormone that is not a tropic hormone or trophic hormone that's released from the anterior pituitary is PRL, which is an abbreviation for prolactin. All right? Now, how do we regulate these molecules? Well, simply put, remember what we said is that one way we regulate them is we're going to be regulating through the hypothalamus. And so the hypothalamus produces a trophic hormone that I thought was just too confusing to call them trophic hormones. So let's call them something more fun. We'll call them releasing hormones, or in some cases they'll be called inhibiting hormones. All right? And surprise, surprise, those two terms are in their names. All right? The other way that we do this is we're going to get negative feedback from the target gland. In other words, what we're going to see is we're going to see a pathway that has 3 steps to it. Now, up here, these are the hypothalamic hormones. Notice how complex this is. Thyroid stimulating hormone, what do you think it stimulates? The thyroid. See how hard this is? And it causes the release of a really, really complex hormone called the thyroid hormone. Actually, there are actually 2 thyroid hormones. It's T3 and T4. It's triiodothyronine and thyroxine are the 2, but we're not going to worry about that just yet. We'll talk about those hormones later. But look what regulates it up here from the hypothalamus. TRH. What do you think that stands for? Thyroid releasing hormone. See, this is not hard. You know, and people who make endocrinology out to be really, really difficult, they're just being mean. All right? ACTH, I said, was the hardest one because adrenocorticotropic hormone. But look what I said. It activates the adrenal cortex. So it's in its name, and the T part is the tropic hormone. And it's being regulated by corticotropic releasing hormone. That's probably the hardest one. GH is growth hormone. It's regulated by growth hormone releasing hormone and growth hormone inhibiting hormone. What it acts on, and this is a little bit weird because you think it acts on any cell and does anything. No, it acts on the liver and causes the release of a molecule called the somatomedins, or molecules. And these somatomedins have another name, the insulin-like growth factors. Why do you think they're called insulin-like growth factors? Because they're like insulin. Yeah, there you go. All right. And then FSH and LH, I said, are specifically called the gonadotropins together. So the molecule that causes them to be released is called gonadotropin releasing hormone. And they act on the gonads, and they cause the release of new hormones, androgens and estrogens. And so actually I failed on it. So adrenal cortex releases cortisol, for example. The liver releases the somatomedins, right? And so here is our second layer of hormones. Remember what I said, the tropic hormones are responsible for releasing hormones in the downstream endocrine gland, so there are your downstream molecules. And then they can act all sorts of places. And then over here, here's that one I said is not a tropic hormone. It's prolactin. It acts on breast tissue. What do you suppose prolactin does? There you go. That's right. Prolactin. For lactation. It promotes milk production. Now, is this all it does? No, but for our purposes and for this class, yes. All right? And then it's regulated by prolactin-releasing hormone and prolactin-inhibiting hormone, which turned out to be dopamine. All right? Now, you can go through and memorize all that stuff and make a nice little chart if you want to. I think that's an easy way to do it. But if you just think about what the molecules do and then think about how do we name these things, you're already 10 steps ahead of everybody who's taking endocrinology. All right? Now, how they all act, we're just going to see that there's actually 2 feedback loops that are responsible for suppressing the pathway in response to activation. So, remember what we said, is that the hypothalamus is responsible for regulating, for turning on a pathway. So the hypothalamus acts on the anterior pituitary. So here's the hypothalamus. It's acting on the anterior pituitary. So whatever hormone the hypothalamus is releasing is going to tell the anterior pituitary to release its tropic hormone. The anterior pituitary, once it releases tropic hormone, that tropic hormone travels to the target tissue, some endocrine gland downstream, causes it to release a hormone. And then that hormone does whatever the function is. So not only does it do its job, but now we have to deal with those 2 molecules. How do we turn off this pathway in response to whatever the stimulation was? Well, that hormone can act in a negative fashion back to both the hypothalamus and the anterior pituitary. In other words, it's a brake to the two things that are turning on the system. And the trophic hormone also can act on the anterior pituitary and act as a brake on the hypothalamus. You can think about it this way. As I'm producing my trophic hormone or tropic hormone from the anterior pituitary, I want to tell the hypothalamus I'm doing a good job. I'm doing what you told me to do. Quit telling me to do it. That's in essence what it's doing, right? That's the negative feedback loop. And then similarly, down here at the endocrine gland, this is saying, again, stop telling me what to do. I've done it. And then I'm going to make sure that you're not nagging me. I'm going to make sure the top boss is not going to nag me. And so I'm basically stopping the process all the way down through a negative feedback loop. And if you learn this pattern right here, you understand the 6 anterior pituitary hormones. Because it doesn't matter what we put here. It doesn't matter what we put here, because the same pathway exists for all 5 hormones plus prolactin. Now I'm going to pause here for a moment. Does that make sense? Good. Yes? That's correct. And so the idea here is that we're regulating things at the level of the hypothalamus. Now, this will make more sense when we talk about the nervous system to a greater degree than what just saying, oh, the hypothalamus does it. We refer to the hypothalamus as the master control gland. It's not a gland, it's nervous tissue. But it controls everything. It controls your body temperature, controls your hunger, controls your thirst. It decides when you need to go to the bathroom, when you don't. Decides basically everything about you. And so it serves as a way to measure what's going on inside the body and then tells the different parts of the body to respond. And so that's why we say the hypothalamus is controlling by releasing its hormone to act on the anterior pituitary to then control things that are downstream. Basically, yes. It's a homeostatic gland or tissue. Tropic is a molecule that regulates the activity or the expression of another molecule from another endocrine gland. I know it's kind of confusing. So A turns on B, B turns on C, C does its work. All right, and A and B were both tropic. Yes, sir. Yes, so they're actually specific cells that are going to be responding to a specific signal with a specific receptor. Now, those particular cells will have names, and we're not going to go any more depth than that, but it's like, for example, the hormone that produces, or the cell that produces luteinizing hormone will have a name called a gonadotrope. FSH will also come from a gonadotrope, but you won't produce LH and FSH from the same gonadotrope. Yeah.
So the anterior portion has exclusively the
Right. That's correct. And that's what we're going to look at here. And it has a real ugly name. Say that after 3 times. Hypothalamic hypophyseal. Hypophyseal. Right. A hypothalamic hypophyseal portal system. And what this is a really, really itsy-bitsy, teeny-tiny, short capillary system between the hypothalamus and the hypophysis, or the anterior pituitary. And what it does is that it allows the body to release this really small amount of message, keep it concentrated so that when it arrives at the anterior pituitary, it's still in its concentrated form. Put another way, your body right now is filled with about 5.5 liters of blood. All right? And normally when you release a chemical message out into the bloodstream, it just kind of travels all over the body. And then hopefully it will find its location, and it usually does. But if you want to send a very specific message, are you going to just send it out into the cloud? No. No. So this has developed so that that message goes directly from the hypothalamus directly to the anterior pituitary. So it has the greatest effect in terms of regulation. You don't have to worry about whether or not that message is going to find its way back. All right? It's right there. All right? So it goes directly to its target, and we're not going to be diluted. Diluted, not diluted. So this is how we get messages from the hypothalamus to the anterior pituitary. Notice how different it is from this mechanism over here, where we have our 2 regions, paraventricular nuclei, supraoptic nuclei, and we are terminating here in the posterior pituitary. Very different systems. Now, I'm going to try to keep this simple and brief to deal with functionality, because I'm probably, if I ask a question, it's going to be simple. I may not even ask a question on this. Again, I have to go over all my questions, and we're coming upon a test in about a week, and you know how that happens. I'm a procrastinator just like you are, and so how many new questions I write and how many old questions I throw out, and yada yada yada, is a mystery to us all. All right, so I say up here the anterior pituitary is referred to as a master gland, and that's actually appropriate. The hypothalamus is the structure that regulates the master gland. All right. And so you can think about this as that the function of the anterior pituitary in real basic terms is growth, metabolism, and reproduction. And we're going to look at these in more details in our very third to last lecture and our second to last lecture ever. So at the end of the semester, we're going to look at these in more detail. But if you can think about what is the anterior pituitary, the three things that are most important to me: growing up, eating, and bow chicka bow wow. All right. Now, the thing to remember, the one thing that stands out are both prolactin and growth hormone have inhibiting hormones. The other ones do not. So they have both a gas and a brake found in the hypothalamus. So there's one feature right there that you can kind of set aside and say only two molecules do this. The rest of them only have releasing hormones. All right. In terms of their functions, prolactin is milk production in females, but plays a huge role in the immune response, which we're just going to ignore for right now. So, whatever. All right, I just want to point out that when you see these hormones, you can immediately think this is its primary function, but you'll find out, if you ever take endocrinology with either Dr. Gill or Dr. Warner, that these molecules are not limited solely by what I'm naming right here. All right. FSH and LH, they regulate hormone activity in the gonads. All right. So they're regulating estrogen and testosterone, plus a couple of other molecules that we'll get to later. So gonadotropins regulate the gonads. Thyroid stimulating hormone regulates secretion of thyroid hormone. Thyroid hormone regulates your metabolic rate. Do you guys know that person that can eat like 7,000 cheeseburgers and never gain a pound? Hyperactive thyroid. Okay? They basically burn a lot of fuel. And so if you hear someone say, I have a high metabolism, thyroid hormone. All right? ACTH, adrenocorticotropic hormone, regulates molecules that are produced in the adrenal cortex. This includes the mineralocorticoids and the glucocorticoids, as well as some sex steroids. I should put that in place, which we'll talk about again a lot later. And then growth hormone has another name. It's called somatostatin, but we're going to stick with growth hormone. Well, it plays a role in regulating metabolism and regulating hormone production through the liver. In other words, the insulin-like growth factors. And again, here's that pathway again. And in this case, we're just throwing in growth hormone so that you can see how it works. I mean, we got growth hormone releasing hormone, there's growth hormone, there's somatomedins, and then what we'd have is we'd have negative feedback here, we'd have negative feedback here, and also we'd have positive feedback in the case of growth hormone to produce what? Yeah, there you go. The growth hormone inhibiting hormone, which would then act negatively. I know that's the one that's kind of confusing right there. I apologize. Now, hormones have all sorts of different types of effects. All right? They have what we refer to as permissive effects. And here what we're trying to say is that the activity of one hormone is going to be dependent upon the presence or activity of a second hormone. And so the example that I have up here is going to be this top example. So basically thyroid hormone actually promotes reproductive development. But it does so in response to or with the help of gonadotropins and sex steroids. And so here we have thyroid hormone. If you just have thyroid hormone alone, you get no reproductive development. But if you throw in steroids and FSH and LH, you get normal reproduction. Similarly, if you just have the steroids and FSH and LH, you're not really going to get good reproductive development. But if you throw in thyroid hormone, you can see how all it works together. This is permissiveness. You have to have all of the players in place in order for things to work. Synergism, simply put, is how these molecules can be complementary to one another. And so I like to do this as a math problem. You all learned in math that 1 plus 1 is equal to 2. But when you're talking about synergism, that's not how it works. 1 plus 1 is equal to 11. All right? So that's what this graph here on the bottom is actually trying to demonstrate. So here you can see the effects of cortisol, glucagon, and epinephrine on blood glucose levels. Each of themselves, they don't do that much. But you put glucagon and epinephrine in at the same time, and you can see a massive difference, a 50% increase in its activity. And then if you put all three of them together, you're seeing a 250% increase. 1 plus 1 does not equal 2. All right? You get these massive effects. It's greater than the sum of its parts. All right? And then lastly is antagonism. And antagonism, simply put, is basically the two hormones fight against each other in terms of their activity. It's kind of a gas and a brake effect. All right? And so the example I wanted to use here happens to be with progesterone and estrogen with regards to pregnancy. Both progesterone and estrogen are expressed during pregnancy. Progesterone's job is to prevent contractions from taking place. But estrogen's job is to force contractions. And so you have to have this perfect balance where during pregnancy you don't get contractions, and so you have higher levels of progesterone, but you need the estrogen for other stuff. Progesterone inhibits the function of estrogen. And then when estrogen levels get higher, they start inhibiting the function of progesterone. And so you're playing kind of a balance game. So that would be antagonism, two things fighting against one another. You can think of other hormones, I'm sure insulin and glucagon you've learned about, which are both antagonistic towards one another. Now, I go over this just very briefly. Any sort of imbalance in hormones is going to lead to some sort of pathology in the endocrine system because of the quantities of hormone that we're actually using. And there are three basic patterns. If you actually produce too much hormone, this is called hypersecretion. Usually what's going to happen is you're going to end up with this massive weird response. All right? And so this is usually the result of either a tumor being present so that you're actually forcing expression, or there's no upstream regulation. And that's kind of the example that's shown in these three slides. So here we have a tumor here in the hypothalamus, which causes an upregulation of corticotropic releasing hormone, which causes an upregulation of ACTH, which causes an upregulation of cortisol, which causes these massive symptoms in response to the high cortisol levels. All right? And there's no amount of feedback that's going to block this because it's the tumor that's producing the hormone. All right? It's not under normal regulation. All right? And you can even take it down to this level. And you can even take it down to this level. And so it's the abnormality, the abnormal expression that's driving the hypersecretion. And no amount of negative feedback is going to stop it. Oop. You also have hyposecretion. This is when you basically have atrophy of a gland or maybe some sort of abnormal trophic hormone secretion through a tumor. This is a little bit different. You may also have abnormal responses as a result of downregulation of the receptor or some sort of failure in the signal transduction pathway. So you can see that it can occur at multiple levels. It's not just a matter of regulation. It can actually occur within the cell and with the downstream targets within the cell, the second messenger systems. Man, so that was an hour of endocrinology shoved in a little tiny box. Are there any questions about this? Does it seem pretty straightforward, not terribly complex? The goal here, I mean, it may feel like it's like, oh my God, there was so much stuff here. But we're just trying to skim across the surface now because when we move forward in these other systems, we're going to start coming back to the endocrinology and these signal pathways. So you can see how these pathways work in the context. Any questions about it? I mean, I know you guys are chomping at the bit. Everyone does about this time. It's like internally in your head, you're just thinking, it's got time to go home. It's time to go home. I got to beat the traffic. Yeah, you're in Houston. You're not beating the traffic. What this leads us into is the other type of signaling. We're going to talk about the nervous system for the last 25 minutes of class, 20 minutes of class, and we're going to bring us to the point where we can understand the action potential and where we can understand the synapse. And that's where we're going to end on Thursday. So, in understanding the nervous system, you got to think about it as a large network of many, many, many nerve cells. And in fact, the term nerve cell is probably not the correct term. Let's just go ahead and just call it what they are. Nervous cells or neuronal cells. All right? Because nerves are a very specific type of grouping of these types of cells. Now, all these cells are going to be linked together in a very organized manner, and their purpose is to control you. Well, not control you, but to control the things that are occurring inside your body. All right? So, the neurons are responsible for carrying electrical signals across their surface very rapidly from one point in the body to the other. And once they arrive at the end where the message is supposed to arrive, they're going to be responsible for releasing that chemical signal, that neurotransmitter that tells the next cell in line how to respond. Now, some cells will take advantage of gap junctions. We've talked about gap junctions. And they'll use these gap junctions to allow for electrical signals to travel between cells. But this is a rather rare case. We see this, for example, in the heart. The heart uses gap junctions to allow the passage and movement of ions so that the heart can pump because all the cells are contracting simultaneously. But neurons, the ones we're thinking about, the ones that are traveling throughout your bodies as nerves and stuff, use neurotransmitters. And you're going to release this neurotransmitter in the extracellular space between the cells. Now there's another type of cell, so we got our neuron, which is cell type number 1. Cell type number 2 are the glial cells. Glial comes from glue, and that's a literal word. All right, this is the type of cells that holds the nervous system together. They're not responsible for directing the transmission of electrical impulses. Instead, they support the neurons. The neurons are the quarterbacks. The glial cells are the line. No one ever gives credit to the line. The thing is, there are more glial cells than there are neurons. About 10-to-1 ratio. Now, the nervous system is organized to make things simple for us, but just because we organize it this way does not make this a compartmentalized structure. The nervous system has both a central nervous component and a peripheral nervous component. The central nervous system includes the brain and the spinal cord. Everything else outside of the brain and spinal cord is peripheral nervous system. So there's your central. Everything else outside that is peripheral. Peripheral nervous system is responsible for bringing information into the central nervous system, which serves as your control center, and then taking the information from the control center back out to create some sort of response to the surrounding environment. So what that means is that the peripheral nervous system has two components to it. It has an afferent component and an efferent component. Afferent and efferent, how's that? Afferent is when we bring information in. That's from the stimuli that's coming from around. And then after we process it, that information is sent away from the CNS through the efferent. So A, control center, back out through the efferent. Now, the afferent basically are a bunch of receptors that are responding to the stimuli. The efferent, on the other hand, breaks down into two different classes again. And in fact, if you go and read your textbook, it's actually going to start talking about a third class that over the last couple of years, they're basically finally saying, you know what, this kind of falls over on the wayside. We're going to ignore that system for a moment. Efferent breaks down into both the somatic nervous system and the autonomic nervous system. Somatic nervous system is responsible for sending signals out to your skeletal muscles, so it is basically your motor neurons. If I step on a crack, or excuse me, if I step on a tack or a nail, I'm going to feel pain. That information, you're stepping on a tack, goes to my central nervous system. My response would be to lift my foot. All right? There's afferent information in, efferent out. Lift my foot up. All right? The autonomic nervous system, on the other hand, does not regulate skeletal muscle. Instead, it's predominantly the glands, smooth muscle, and cardiac, which is now hidden underneath a bunch of red. These are the things you do not control voluntarily. All right? You cannot control your heartbeat. I know you think you can. I know you think you've seen shows where people can do this. You know, like the guy who gets shot in the movie and then pretends like he's dead and then gets up and saves the day. No, does not happen. All right? You cannot save yourself. I mean, I'll prove it to you. Everyone, I want you to make your heartbeat go faster now. Go. Do it. Thinking about it doesn't happen. Now, if I opened the doors and said, here's a lion, would everyone's heartbeat go up? Yes. All right? So it's your response to the stimulus, and it's a natural response. Say, it's not voluntary. You can't say, I'm not going to make my heart beat faster when I see that lion, especially when he starts looking at me and doing that. Now, in the autonomic nervous system, we can classify neurons within this into two different categories. We classify them both as the sympathetic and the parasympathetic. And we'll deal with this a little bit more after the exam. All right? But my point is, in all of this division and stuff, is that this is just a way to make it easy for us to deal with like location and structures. It's an integrated system. And so we can't just focus on one and just pretend like it doesn't affect other systems and stimuli is only coming through over here and it doesn't have an effect on the downstream side. No, this is all integrated together. So this is just humans like to classify things, biologists especially like to classify things. This is how we've done it. The neuron, the cell that does the electrical signaling of the nervous system, actually has a whole bunch of parts to it. And you might be familiar with these, but just in case you're not, we have the cell body. The cell body is where all the organelles are going to be located. Then we have dendrites. This is where we receive signals. All right? So at this point, this is where we're going to receive some sort of stimulus, which is going to result in the production of what we refer to as a graded potential. The stimulus gives rise to what we call a triggering event. All right? And then, moving away from the cell body, the long dendrite is called the axon. All right? And this axon is where we're going to send an electrical signal called an action potential away from the cell body to the axon terminals. Now, the place where the action potential is going to be generated, and again, if you don't know what the action potential is, don't worry, we'll talk about it tomorrow or Thursday. The action potential is going to be generated at a structure called the axon hillock, which is simply put the point where the axon and the cell body meet. All right? The axon terminal, this thing right down here on the very edge, is where that action potential is going to be terminated, and it's going to cause a release of a chemical at that terminal. And notice what it does. In our little cartoon here, which is a classic example, is that that terminal terminates right next to the dendrite of another neuron. And so what we're doing is we're releasing chemical and we're influencing the next cell at the exact same place where we said we're going to receive triggering events. So we're going to be kind of playing a chicken and egg thing here. Which comes first? All right? Now, there are three different classes of neurons. You can break them down into afferent neurons, interneurons, and efferent neurons. Afferent neurons are structured in such a way, using our example, they have an end that is called a receiving end or a receptor end. And so usually in this area you have receptors of some sort that detect a very specific type of stimulation. All right? Then what we have is a very long body, long axon. And actually what this is actually two dendrites that have actually fused during development. So you end up with a cell body that sits off to the side. The axon actually looks like one very long axon with the cell body sitting off over to the side. And so when stimulation occurs down here at this receiving end, an action potential is going to be sent along the length of that long axon down to this little short axon, which is then going to enter into the central nervous system, and it's going to terminate on interneurons. Now notice, if I say, here is my peripheral nervous system, here's my central nervous system. What are the things we can say about the afferent neuron? The cell body is located in the periphery. All right? The receiving end, the afferent portion that actually receives the stimulus is in the periphery. And that information is then sent into the central nervous system. So the majority of our our cell lies within the peripheral nervous system, which kind of explains that whole afferent neuron stuff, or afferent component of the peripheral nervous system. Interneurons lie completely within the central nervous system. And they're called interneurons because they lie between the afferent and the efferent, which kind of makes sense. All right? When you think of neurons in the brain, this is what you're thinking about. They make up 99% of the neurons in your body, and their count is roughly about 1 times 10 to the 11th number of cells. So there's a lot of them. All right? And their job, simply put, is to integrate information. I receive a stimulus. How do I respond? If I step on a tack, what should I do? All right? I'm being chased by a bee. How do I behave? I see a beautiful woman. What do I do? I see a cheeseburger. How do I respond? All right? It deals with taking stimulus and responding to it. And then the other thing that it does, and this is the part we don't get. We haven't figured this stuff out yet. How does it regulate, or how does it deal with our abstract mind? You know what I'm talking about, right? Thinking. I mean, you guys have been daydreaming for, 30 minutes now? You know, how does it come up with stuff? You know, how does it store memories? You know, these are the things that we don't understand about the interneurons. And then the last portion are the efferent neurons, and these lie predominantly, again, in the peripheral nervous system, but its cell body is located within the central nervous system because the interneurons have to come into contact with those peripheral ends. All right? And so a signal that comes in through the through the afferent is going to stimulate the interneuron, which then stimulates the efferent neuron, and you're going to get some sort of response down here. It's an effector. All right? So again, if I step on a tack and I detect the penetration of that tack through the bottom of my foot, that's going to come into my central nervous system. Notice what we have here. It says spinal cord. Don't think it's all up here in the brain. It can occur at the level of the spinal cord. Step on a tack. Ow! Lift my foot up. I'm telling my muscle to do that. If I smell a wonderfully cooking cheeseburger, what's my response? Salivate. All right? So the effect is a result of the efferent neuron, the activity that's done down. Glial cells, as I said, are the glue. And there's a whole bunch of different types. They don't conduct nerve impulses. They serve as the connective tissue of the brain. And so their job is to maintain the extracellular environment. And so this is where it gets really fun because there are 4 types in the central nervous system and 2 types that are found in the peripheral nervous system. They have really weird names, and function-wise, you're kind of like going, I'm not sure what is what and who is who. Well, simply put, we named them for what they look like, and then we figured out their functions afterward. Astrocytes, they look like stars, and their job is they are responsible for, and you can see a picture of, actually I have a better picture. Their job is to basically provide both physical support and chemical support. So you can think of a star has 5 points. There, I'm an astrocyte. I'm supporting things. All right? I'm holding up a neuron over here. I'm holding a neuron over here. I'm holding a neuron down here. All right? Oligodendrocytes. Oligo means many. Dendrocyte means many dendrites. So it looks like it has a whole bunch of dendrites. Their job, and you can look at it here, where is my oligodendrocyte? They basically have a whole bunch of extensions that go off in all sorts of different directions, and they form these myelin sheaths. We'll talk about what myelin does in the nervous system a little bit later. Now, the counterpart to that oligodendrocyte in the peripheral nervous system is the Schwann cell, which is easy to remember because I always think of Schwinn. I don't know why. It's kind of look alike. All right, and they form myelin sheath. They just do it differently, and we'll go into a little bit more detail about that. Microglia, small glial cell. Their job is immune defense. When you get an infection in the central nervous system, their job is to respond. And lastly is the ependymal cells. Ependymal cells, they line the central cavities of the central nervous system, which will make more sense when we talk about it, and they produce the extracellular fluid and the spinal fluid of the nervous system. This picture just kind of tries to put things into perspective, and you can see the microglia just kind of cruising around over there. Here's the oligodendrocyte. It's wrapped itself around here and wrapped itself around there to put myelin sheaths around our neurons. You can see the astrocyte, which is basically just creating support. Here's another one, creating support, so on and so forth. They really are actually rough. They're more or less inactive until there's some sort of insult. But they're in that space. Yeah, they're kind of like the macrophages in your skin. If you didn't know you had them, they just kind of sit there and they wait for an insult. And when an insult occurs, then they become activated. And when I say insult, when they get exposed to an antigen. Yes, I'm looking at the clock. I know what's going on here. See, if I stop here, it's going to make Thursday hell. So it's up to you. Do you want me to stop here? I see the heads going, I could do 3 more slides, or really my goal was, I think, 4 more slides. But I don't think I can get through 4 slides in 5 minutes. She's looking at you. I thought it looked like, no way. Let me just set the stage for Thursday, all right? We won't even get into the graded potential, but we'll talk about what we're going to get to, all right? And really what we're going to try to talk about is how we go about producing an action potential. We've talked about how ions move back and forth across membranes. We have a membrane potential, and what we want to do is we want to turn that membrane potential into a signal, all right? So what we're talking about is the inward and outward flow of these ions, all right? Now, one of the things we need to understand when we're talking about these movement of ions is that we're really not talking about a lot of molecules. We're talking like 1 or 2 molecules that cause these massive changes, about 100 millivolt changes in membrane potentials. Now, remember what we said is that in order for ions to pass back and forth across a membrane, we have to have channels that allow them to do that. All right? Now, the channels that we're going to be looking at are gated channels. In other words, these are channels that are going to be closed and need to be stimulated to be open. In fact, this is how we detect the environment around us. We have to have some sort of stimulus that results in the opening of these gates. So there are many different types of gates. I only threw up 3 up here. We've got voltage-gated channels, and these are channels that open in response to changes in the membrane potential. So they're actually kind of responsive to when ions start passing through, voltage-gated channels open up and allow more ions to pass through. All right? We have chemically gated channels, which is the easiest one. You can just think of a chemical coming along, binding to the receptor, causing the channel to open up, and it allows something to pass through. All right, this is going to be really obvious when you see them. Mechanically gated channels respond to stretching or other kinds of mechanical deformation. Everyone, I want you to grab your pen real quick, or pencil, and I want you to push it against your skin. Do you see mechanical deformation? Yeah. You see the skin indenting, right? Well, guess what? The reason you can feel that pen is a responsiveness to those mechanically gated channels. In other words, what you did is when you press down, you caused the cells to change their shape. And when they change their shape, that was detected because of the receptors that are found in the surface or within the particular receptors that have those channels. So each of these different types of channels have different thresholds. This is kind of why you can actually sit there and like poke yourself and get like a molecule of the pen touching you and you can't feel it. But then if you start pushing down, you can actually start feeling it. It's because there's a threshold that results in the opening of those channels. All right? The other thing that's really important to understand before we move forward is that these different channels open and close at different speeds. All right? So it's kind of like you can open 2 channels simultaneously. You know, different ion channels, but one opens fast, one opens slow, so it appears like they're opening in sequence. Like one's opening and then closing, and then the other one's opening and closing. All right? So the example I want to use here is that sodium channels are ones that open very, very fast, whereas potassium channels open slowly. We're going to look at the action potential where both of these channels are stimulated to open at the same time, but one occurs before the next. Yeah. No, this is just a function of its structure. Yeah. And then when you close the gated channel, it can be the result of 2 different things. Either you're repolarizing the membrane, or you're getting some sort of inactivation of it. Now, when we come back, what I want to talk about is we're going to talk about the types of potential change that result in signals being sent across the cell. And what we're going to be talking about are graded potentials and action potentials. And how we get those is basically through the flow of very specific ions. And we'll see how fast I have to talk on Thursday. I shouldn't have to talk fast.
Is log mass action